Welcome everyone to this week's edition of the Extracellular Vesicle Club. This week, we are devoting the EV Club to the discussion of tissue factor. And so the importance of tissue factor is very hard to overemphasize. And we're going to be learning today about how tissue factor is associated with small and large EVs and in different diseases. And for this discussion, we're privileged to have on hand Nigel Mackman and Anna Sachetto from his lab, as well as Rink Newland. So Rink, thank you so much for recommending this article for EV Club. I'm very much looking forward to learning more about tissue factor today. And Nigel, I would like to hand this over to you in just a second. But first, let me remind everybody that during the presentation, you can put your comments and questions in the chat box. And then at the end of the presentation, we will allow unmuting. We'll allow you to show your camera if you'd like to, and you can then discuss with Anna and with Nigel and Rink about the paper and the details. So Nigel Mackman, thank you so much, and please, it's all yours.
So just a way of background, I've been working on tissue factor and blood clotting for 35 years. I've been working on extracellular vesicles for 15 years. I'm more interested in their procoagulant activity. So we developed a lot of assays, and Anna joined the lab last year and has done a fantastic job now taking on studies on EV tissue factor. So Anna, it's all yours.
Okay. First of all, as Nigel said, I'm a postdoc at his lab, and I want to thank first Ken and organize for this opportunity, and thank Rink for inviting us and suggesting our paper. So I'm very excited today to talk to you all about tissue factor activity of small and large extracellular vesicles in different diseases, and thank you for everyone for being here today. So I'm gonna talk a little bit of a quick background about procoagulant EVs, and then go in a little more detail about the measurements of phosphatidylserine positive EVs and tissue factor positive EVs, and then get into the results from the paper about the tissue factor activity of small and large EVs in different diseases. So as a first step or background, the activation of coagulation leads to the formation of clots, but these clots are different and have several types. So on arterial thrombosis, these clots are mainly platelet rich, and on venous thrombosis, they are mainly fibrin rich, and they all possess blood cells and fibrin. So there's a few requirements for blood clotting to occur. One of them is the presence of an activator. So it's tissue factor for the extrinsic pathway of coagulation cascade, or negatively charged surfaces for the activation of factor 12 and the intrinsic pathway. We also need calcium ions for the reactions to occur, clotting factors that are present in plasma, and also phospholipid surfaces, which are mainly activated platelets or EVs present in plasma, and also fibrinogen. So in a simplified way, the extrinsic pathway of the coagulation cascade is activated upon a vessel injury, and the intrinsic pathway is activated by negatively charged surfaces. So in vivo, tissue factor is the main initiator of the coagulation cascade, and it forms a complex with factor 7A. This complex activates factor 10A, factor 10, and the formation of a complex with factor 5A then activates thrombin, which is a key protease in this cascade. So thrombin activates platelets, also creates a feedback amplification loop for the coagulation cascade, and importantly, transform fibrinogen into fibrin that later on forms a cross-linked fibrin net. So more specifically about tissue factor, it is a transmembrane glycoprotein, and it's a receptor for factor 7, factor 7A factor 7 activated. And as I said before, it is the primary activator of coagulation. Furthermore, this complex of tissue factor effector 7A is essential for hemostasis, but it also contributes to thrombosis. So besides an initiator as tissue factor, phospholipid surfaces are also really important for clotting to occur. And on a plasma membrane of eukaryotic cells, there's a phospholipid asymmetry. That means that positively charged phospholipids, such as sphingomyelin, are usually located in the outer leaflet of the membrane, and negatively charged phospholipids, such as phosphatidylserine, are usually located in the inner leaflet of the plasma membrane. But the loss of this asymmetry in damaged cells can lead to phosphatidylserine exposure or negatively charged phospholipids exposure, and that can increase clotting. So that occurs because many clotting proteins, such as factor 7, factor 9, 10, and prothrombin, contain a positively charged glad domain, and this domain binds to negatively charged phospholipids, such as phosphatidylserine. So when we have the loss of this phospholipid membrane asymmetry, phosphatidylserine that is then more exposed, and that can lead to more assembling of clotting factors on the membranes. So the contribution of EVs to thrombin generation in plasma are studied since the 40s, and on 1946, it was observed that plasma freed from intact platelets generated thrombin, and that this thrombin generation could be reduced by prior high-speed centrifugation of the plasma. So this was the first observation about that. Following that, in 1967, it was observed that the majority of EVs in blood of healthy individuals were derived from platelets, and they were originally described as platelet dust. Early studies then reported a wide range of platelet-derived EVs in blood of healthy individuals by flow cytometry, and more recent studies reported a presence of almost 100,000 EVs per microliter of plasma from healthy individuals. So these studies were really the basis for the further studies that were done regarding procoagulant extracellular vesicles. And now we know that many diseases are associated with increased levels of EVs in circulation, and that these EVs are really a very heterogeneous population in blood. So they have different activities, sources, and sizes. So the procoagulant EVs are mainly phosphatidylserine positive and or tissue factor positive. So the PS positive EVs are mainly released from red blood cells and platelets, and they have a tissue factor independent procoagulant activity. The other EVs that have both phosphatidylserine and tissue factor are mainly released from monocytes or tumors or cancer cells, and they present a tissue factor dependent procoagulant activity. Okay, so now that we know that these procoagulant EVs are important, let's understand a little bit more about how to measure them. So there are two commercial assays to measure levels of PS positive EVs in plasma. One of them is a functional assays, where the PS positive EVs are captured onto plates using an Xen5, and the assay measures the activity of the prothrombinase complex right here. The second commercial assay is based on the principle that PS positive EVs would provide a surface for clotting in a phospholipid depleted plasma, and then the clotting time would be a measure of the levels of PS positive EVs. So a little more in depth about these assays, the Zymofin MP activity assay is done by coating ELISA plate with streptozidine and biotinylated Xen5, And after a wash, the diluted plasma is added containing thrombin and factor Xa inhibitors. And then that allows the PS positive EVs to bind to annexin 5. And then after another wash, factor Xa and factor 5 and prothrombin are added, and the formation of the complex is allowed on the PS positive EV surface, and that allows the conversion of prothrombin to thrombin. And then this thrombin is then measured by the cleavage of a specific thrombin chromogenic substrate. So several studies used this assay to measure PS positive EVs on plasma or some human samples, and they showed that circulating EVs are increased in patients with acute kidney injury and also patients with endometriosis. And there was also shown that elevated levels of circulating EVs are independently associated with reduced risk of acute respiratory distress syndrome in critically ill patients. The second commercial assay for measurement of PS positive EVs uses phospholipid depleted plasma, and this is mixed with the test plasma or the sample. And then factor Xa and calcium are added, and clotting time is measured. But this commercial kit requires an automated analyzer. So again, some studies use this commercial assay to measure EVs, and they show that circulating EVs are increased in patients with essential thrombocythemia and also during pregnancy. And importantly, elevated levels of circulating EVs might increase the risk of thrombosis in patients with thrombophilias. So about the tissue factor positive EVs, we know that healthy individuals have very low levels of TF positive EVs in blood. However, activated host cells and cancer cells can release tissue factor positive EVs. And they were shown to be elevated in several diseases, including cancer, bacterial and viral infections, endotoxemia, sepsis, sickle cell disease, and also liver injury. And importantly, tissue factor positive EVs are associated with thrombosis and disseminated intravascular coagulation. So there are several functional assays to measure tissue factor in plasma. They can be done using plasma, EVs isolated using centrifugation, or some of them use the capture of EVs onto plates or the capture of EVs onto beads. They are mainly divided in 3 categories. So these assays would analyze the generation of factor 10 activator, or the generation of thrombin, or the fibrin formation. Very importantly, factor 7A can directly activate factor 10 independently of tissue factor, and therefore there's a need of induction of inhibitory anti-tissue factor antibody. So this is required to distinguish between the tissue factor dependent and tissue factor independent factor Xa generation. And according to that, a recent study by the Vascular Biology Scientific Standardization Committee of the International Society of Thrombosis and Hemostasis evaluated 18 assays for the ability to measure EVTF activity in plasma. And only the assays that include this anti-tissue factor antibody were able to reliably detect tissue factor positive EVs, showing the importance of really distinguishing tissue factor dependent and independent activities. So this is one example of the methods to measure tissue factor. This is our in-house factor Xa generation activity assay. So EVs are isolated using centrifugation, and these resuspended EVs are then added to the plate. And incubated with an anti-tissue factor antibody or a control antibody, so we can distinguish between tissue factor dependent and independent factor Xa generation. After that, the samples are incubated with factor 7A and factor 10 and calcium, and then incubated with a factor 10A substrate to analyze this generation. And for that, we considered 0.5 picograms per ml as the background of EVTF activity in this assay. So it really is our threshold for that. And also the TF-independent factor 10A is a measure of levels of phosphatidylserine positive EVs in this assay. Okay, so using this assay, we analyzed the levels of EVTF activity in patients with COVID-19, and we observed elevated levels of EVTF activity in these patients compared with healthy controls. And the levels of EVTF activity were associated with the severity and mortality in COVID-19 patients. Another study analyzed small and large extracellular vesicles in COVID-19, and for that they used differential ultracentrifugation to isolate large and small EVs. And for that, they considered that the large EVs were the ones that were isolated using 20,000 G centrifugation, and the small EVs were the ones considered isolated with 100,000 G centrifugation. So they analyzed these samples enriched for larger and small EVs using TM, and observed a larger size EVs in the large EV samples and a higher number of smaller size EVs in the small EV samples. So showing that this method of differential centrifugation was in fact effective in distinguishing these two populations. So importantly, they analyzed the procoagulant activity of large and small EVs from COVID-19 patients. However, they used a commercial kit without an anti-tissue factor antibody. So in reality, they measured just the total factor Xa generation and not the tissue factor dependent Xa generation. But this study was really important to us as a kickstart to think about the contribution of small and large EVs in the tissue factor activity in several diseases. So thinking about that, we did this study specific about tissue factor activity in the small and large EVs in different diseases. And for that, we had amazing collaborators, including RINC, and that allowed the observations of different samples from different diseases. So the main objective for this study was to analyze tissue factor activity in two EV populations enriched either for either large EVs or small EVs in patients with sepsis, pancreatic cancer, or COVID-19. For that, we used different samples. So first of all, we did positive and negative controls for tissue factor positive EVs. So blood was collected from healthy volunteers and plasma was obtained, and that was considered or negative controls. And this blood was also incubated with LPS, which is a known inductor of tissue factor, and then processed to obtain plasma, and that was considered or positive samples or positive control samples for tissue factor positive EVs. We also used plasma from patients with cancer, sepsis, or COVID-19, and these samples were selected from previous studies from our group and selected so we could have a range of EVTF activity. So the EV isolation started with a centrifugation of 20,000 G, and then the supernatant and the pellet were separated. The pellet was then resuspended and centrifugated again at 20,000 G, and this pellet was considered or samples enriched for large EVs. After this first centrifugation, the supernatant was then centrifuged as at 100,000 G twice, and this palette was then considered as our samples with enriched for small EVs. So both samples, small and large, were then analyzed with our factor Xa generation activity assay using anti-tissue factor antibody. Okay, so first of all, we analyzed our positive and negative control samples. And we observed that EVTF activity was increased in all samples with LPS compared with minus LPS samples in large EVs, but not in small EVs. And we observed before in other studies that the stimulation with LPS increases EVTF activity in large EVs. So this is congruent to previous studies. And we also observed that EVTF activity in plus LPS samples is mainly due to large EVs. So again, just to highlight, our threshold of 0.5 picograms per ml will be used as our limit of detection for this assay. So these results are congruent also with another study, another previous study from our group, where EVs were isolated with a 20K G centrifugation or 100K G centrifugation, and similar levels of EVTF activity were obtained with these samples. We also analyzed the tissue factor independent factor Xa generation, so the one that is associated with the presence of PS positive EVs. And we observed that LPS slightly increased tissue factor independent procoagulant activity in large EVs, but not in small EVs. And we also observed higher levels of tissue factor independent activity in small EVs compared with large EVs. When we analyzed the sepsis patient samples, we observed that 2 out of 17 patients had EVTF activity in large EVs, so the ones that were above our threshold or background threshold for the assay. And this is congruent also with a previous study where we showed that 4 out of 35 patients showed TF activity in large EVs. But we also observed that 2 other patients had EVTF activity in small EVs. However, they were not the same patients that had increased levels of EVTF in large EVs. We also analyzed the TF independent procoagulant activity in the samples. And we observed that a few patients with sepsis had elevated levels of tissue factor independent procoagulant activity in both large EVs and small EVs, but not necessarily the same patients. Moving on, we analyzed the pancreatic cancer patients, and we observed that 6 out of 10 patients had EVTF activity in large EVs, and the same patients had EVTF activity also in small EVs. And very importantly for us, we observed that EVTF activity was actually higher in the small EVs compared with the large EVs. Analyzing the tissue factor independent procoagulant activity, we observed that 6 of the patients had higher levels of tissue factor independent factor Xa generation in large EVs and small EVs compared with the other patients. And the same patients actually had increased levels of EV tissue factor activity. Last but not least, we analyzed the COVID-19 patient samples and observed that 6 out of 12 patients had increased levels of EVTF activity in small EVs compared with the other patients. And also 6 out of 12 patients had EVTF activity in small EVs. And importantly, they showed, most samples showed similar levels of EVTF activity in large and small EVs. When we analyze the tissue factor independent procoagulant activity in the samples, we observed that in general, the patients had increased levels of EVTF activity, showed increased levels also of tissue factor independent factor Xa generation in both large and small EVs. So our main conclusions for this study was that the procoagulant EVs are released into the circulation by a variety of diseases, and they likely contribute to the pathological activation of coagulation in these diseases. Specifically in pancreatic cancer patients, the levels of EVTF activity in small EVs were higher than large EVs. And in COVID-19, most patients with EVTF activity in large EVs also had EVTF activity in small EVs at similar levels. So importantly, we recommend using 100,000 G to isolate EVs from plasma to measure their procoagulant activity, so more accuracy is obtained. And as future studies, we understand that future efforts should attempt to standardize the isolation of EVs for the measurement of tissue factor in plasma, and further studies are required to evaluate the potential of EVs isolated using a centrifugation with 100,000 G as a biomarker for pathological activation of coagulation, and also possibly a biomarker for thrombotic events. I want to thank the Blood Research Center, specifically the Mackman Lab, also our collaborators, financial supporters, and our collaborators for this study specifically, including RINC. So thank you.
Thank you very much, Anna, for that excellent presentation. We're looking forward to having some good discussion here with the audience, and I am currently allowing people to unmute themselves, share their videos if they like. So please join our speakers or our speaker and then our PIs here too. So I will call on people who have put a question in the chat box and invite you to expand on your question. And let's see here. So we already have a couple of questions in the chat box, and everybody else who hasn't put their question there, just put them in the chat box so that we know whom to call on. So let us begin with Wolf Hontoner. Please go ahead.
Yeah, thank you very much, Ken, and thank you very much, Anna, for your very nice presentation. Very, very interesting. To be honest, tissue factor positive EVs were back then, long time ago, where I stepped into the EV field. So I was wondering about the origin of the tissue factor positive EVs. I mean, you were working with plasma samples, right? And we all know that the way majority of the EVs in plasma are derived from platelets. So do you know about the origin of these tissue factor positive EVs, or do you know whether they come from different cell types, or if they even come from the vasculature? Because this was our project back then. We analyzed EVs from cultured endothelial cells and found if they are stimulated with pro-inflammatory stimuli like TNF-alpha, they exert tissue factor pro-activity. So
okay,
could you comment on this?
For sure. So in our disease models, the tissue factor positive EVs are usually related with monocytes or cancer cells and tumors. But so I think that's usually.
So it's a very difficult problem to address because the levels of tissue factor in the circulation are extremely small. And we really don't have the tools. We can't detect tissue factor in plasma by flow on the vesicles. And so we can't do co-staining with cell-specific markers. We believe in pancreatic cancer, the majority of the vesicles are coming from the cancer itself. And if you resect the cancer, the levels go down. So there's a correlation in terms of levels versus the size of the tumor. In terms of the COVID, the only work that's been done is by Francois George in Marseille, who took COVID positive samples and then depleted monocyte-derived vesicles, and the level of EV tissue factor went down, suggesting that at least a portion of those tissue factor positive vesicles in the circulation of COVID patients are derived from the monocytes. And that's consistent with data showing that monocyte tissue factor expression is increased in COVID patients. But we really don't have the sensitivity. We're trying to develop techniques to look at that, but at this point, we haven't come up with an ability to try and detect the cellular origins of those tissue factor positive vesicles.
Okay, that's very interesting. Thank you very much, Nigel. But what do you think? What might be a solution for this? Do you think you can fish out by antibody-coupled magnetic beads or whatever specific EVs which are not that abundant in the plasma?
Anna, do you want to?
Yeah. So we have been trying, and we're still trying to figure it out ways to be able to detect and analyze better this TF positive EVs. So we tried magnetic beads. We tried isolating them with centrifugation and then running Western blots. We also already tried before using flow cytometry, but so far none of these methods were really, let's say, useful or effective in this analysis. So we're still looking, still searching, and still trying to find a way to analyze this tissue factor positive.
Okay, maybe last question: which antibodies did you use for flow cytometry?
Many different antibodies, and in fact, a sort of follow-up of the SSC study is that there were 9 different labs that claimed they could measure tissue factor by flow, and we gave them positive, negative samples. RINC was involved in this, and all failed, completely and utterly failed to detect tissue factor on the vesicles with a variety of different anti-tissue factor antibodies. So I will say that the group down in Marseille worked with Stargo to try and commercialize this assay, and they've developed a bead capture system where they can capture the tissue factor positive vesicles onto beads, and they increase the sensitivity somewhat. And the idea would be potentially to try and collect them onto different cell type specific beads, and then sort of co-determine whether the activity is associated with tissue factor. But it's really, the main limitation is the extremely low levels of tissue factor. And if you have high levels of tissue factor vesicles in the blood, you're dead. So that's not really a good option.
Okay, so taken together, in your opinion, it's a point of sensitivity, right?
Correct.
Yes.
Yeah.
Correct.
Yeah. Thank you very much.
Thank you.
Thank you all for the questions. Next is Graciela Cricun. You have a question about a specific condition.
Yes. I am particularly interested in EV production and tissue factor by EVs from endometriosis, which is not malignant, but it has a lot of the same properties as cancer. And you mentioned that you looked at that, and I was wondering if you published that study.
Nigel,
Yeah, so we haven't looked at endometriosis. The only reference to endometriosis was that the Zymufin assay. There was a publication on increased levels of PS positive vesicles in endometriosis. If you had the samples, we would be more than happy to measure tissue.
We spoke before about that.
Yeah,
and,
yeah. So,
unfortunately, I haven't been able to collect,
right.
Samples, plasma from patients with and without endometriosis.
We're a really good basic science lab. We have to rely on collaborators to get the clinical samples, which is why I think the interesting thing about Anna's paper was not that we could measure tissue factor in big or large vesicles from LPS stimulated blood, but the differences in the different diseases.
Yeah.
And I think it really opens up a whole different sort of set of questions is, you know, why are there different sized vesicles containing tissue factor? Where are they being produced from? And I think that sort of is what we would like to understand going forward.
Well, Nigel, I am going to keep trying to collect the samples, but the second question I have is, all the vesicles that you isolated were from by centrifugation, ultracentrifugation. I have been using kits to isolate the vesicles. Do you think that that works as well, or do you think it shouldn't be done that way?
Anna, you can talk about Josh's data.
Yeah, I think it can work and depends on the sample. So Josh's data that Nigel mentioned was using ExoView, and that worked for some samples from LPS or LPS positive and negative controls, but it did not work for clinical samples from trauma patients and also variant cancer patients.
So the bottom line is you recommend ultracentrifugation versus kits?
There's also a limitation that some kits that isolate EVs could affect the EVTF activity assay. So there's also another limitation, but I think it would be interesting to maybe test different methods of isolation of EVs, and that's actually something that we want to pursue in the future.
Okay.
So I know Dr. Borelli's on the call here, but we, in our view, we have failed to detect tissue factor antigen levels on vesicles, and we have to use functional assays to measure because of the lack of sensitivity. And we found 10 to a 50-fold increase in sensitivity using a functional assay versus an antigen-based assay. And again, with the ExoView, which is exquisitely sensitive to capture these tissue factor positive vesicles, we found our positive controls work great, but then we went into clinical samples and we just couldn't get a signal above the background of the assay. So again, I would recommend functional assays to measure tissue factor vesicles and not antigen-based assays, which have low sensitivity. And also there's debates about which antibodies that I know Bjarne Osterud is on this Zoom, and he can give you a whole lecture about specificity of anti-tissue factor antibodies if you'd like to hear that.
Okay, so let's say that I incubate stromal cells, which are the ones that, you know, show tissue factor, and if I incubate them on the normal control medium, or maybe with TNF alpha, which is, or NF kappa B, which is well known to induce an endometriosis-like environment, if I could isolate those vesicles from the two conditions, could you maybe use that to?
Yes, we do culture supernatant regularly. So we usually have culture supernatant from pancreatic cell lines.
All right.
We have protocols for isolation from the supernatant and can measure vesicle tissue factor.
That's great. Okay, then I'll do that. And
That's great.
okay,
Yep.
That's great. And I'll email you. Okay.
Yeah.
It's nice to see you again.
And you.
We both got older.
Thank you, Graciela. This I think is a very important set of points that have been brought up about the separation procedures that we use and what effect they might have, you know, depending on the matrix that the EVs are contained in too. So we've heard that with the clinical samples, sometimes the results are a little bit different for something like the SP Iris platform. And in fact, we have another comment here before I go to anybody else, a question. So we're going to go a little out of order here. So Young is asking a question about separation specifically. So Young, would you like to go ahead and ask that?
Hi, nice presentation. I'm from Korean Clinic, and my name is Young. So I'm kind of curious about, you know, isolation method you used. Have you tried to, you know, isolate EV using size excluding column? Because, you know, 100,000, you know, centrifugation may disrupt, you know, EV structure. So any negative effects on tissue factor activity assay?
We thought about that, but because of our limitations of the volume of samples that we had, that was we couldn't use the columns to do that. But it would be interesting for sure to use that and compare in the future.
Okay.
So you know, this is probably not the right audience to say this, but we really don't care about the structure of the vesicles. We just care about their procoagulant activity. So whether they're disrupted or not disrupted, we're just measuring their procoagulant activity. I entirely agree. You know, if we had larger volumes of sample, we would use size exclusion and treat them more delicately. We're using the term EV tissue factor activity assay in a sort of broad sense. It's basically anything that you can spin out of plasma. And we actually, when we did initial studies, looked by EM of what we produced, and there were a lot of products being, a lot of sort of debris as well. So these are not highly enriched, purified vesicles. We're just studying the procoagulant activity in the circulation that could potentially be thrombotic and contribute to thrombosis in different diseases.
Right, agree. Okay, thank you.
Yeah, excellent points. And I see that Tatiana has also put a comment in here. So Tatiana, feel free to unmute yourself and jump in here, but you probably heard about the...
Yeah, I already see that my question was a bit late on that topic, but yeah, there are a lot of different isolation methods that one can still consider. So if we want to set a goal for higher purity, maybe I understand that there is a sample volume limitation, but maybe in future work, there's also other potential purification and isolation methods that may be worth considering.
For sure,
Yeah.
that'd be really interesting moving forward.
Yeah, and we can see here how that, you know, different groups can have different questions about the same samples, right? So I'm hearing strongly that, you know, the important thing that's being investigated in these studies is really the activity. So whether it's associated with in this way or that way with an EV is not so important if you just want to understand what the activity is. Whereas there might be another question that asks, how exactly is this associated with EVs? How is the epitope perhaps masked by other factors? And so can we use different separation techniques to get to the heart of that that association? So all good suggestions.
I mean, I would make one comment that there are huge numbers of platelet-derived vesicles in plasma. There is essentially zero tissue factor positive vesicles in plasma. So we have a much easier time in terms of distinguishing the elevated levels versus background. I think when you talk about total levels of vesicles and platelet-derived vesicles, you have a very high number to start with, and then you have to potentially see a higher number with a disease, and sometimes it's very difficult to go above that high number because of the background levels in a healthy individual. So at least we have that as a benefit. And I'm looking at this question about filtering with the cutoff of platelet EVs. Platelet EVs don't contribute to our tissue factor dependent activity.
Avital, does that answer your question, or do you want to follow up?
Yes, it does.
Okay, very good. Thanks for that. All right. Yeah, on the topic of platelets, are platelets cells or EVs?
Great question.
Sorry, it's a sore point.
They're definitely a cell. Small.
Rank, do you agree?
Small cell.
It's interesting. I really would like to make a little survey, a little questionnaire, and send it to platelet experts and EV experts, and to see what they would answer, and write a little paper about it, because I think there's really no consensus there.
I think that would be perfect, actually, because, you know, yeah, depending on your perspective, you get a very different answer, and the answers too are they're so informative about what is important to, you know, individual research projects and individual investigations into physiology and pathology and so on. So yeah, it's interesting. So sorry for that.
I mean, I think one point to distinguish that's important is that tissue factor positive vesicles will initiate blood clotting, whereas PS positive platelet-derived vesicles will propagate ongoing clotting but cannot initiate clotting. So that's an important distinction in terms of their functional activities.
All right, so I did skip over a few questions here because this person Jayachandran had to leave, and it made sense to go to the other one too. But let me see if I can read these out. How do you separate non-EV associated tissue factor thrombin generation? That is to say, and I guess what we've heard over the last few minutes is that it's not, you're pelleting things. It could be that there are some things that are not associated with the EVs, correct?
Yeah, it could be, but in our assay, since we isolate the EVs first and then resuspend and use these EVs, then it's what we say it's all associated with EVs.
No, you can't see that. That's exactly the point. We're spinning down anything from plasma. We don't know the proportion that's EV associated versus cell debris associated.
Oh no, cell debris, yes. I thought
So that's still going to be in the assay.
Soluble forms.
Right.
Excuse me.
Yeah.
And maybe it's good to mention that the tissue factor is a transmembrane protein. So it is not, if it is really associated with a double phospholipid bilayer. It is not present, I don't know, just it needs to be present in a vesicle or a remnant or whatever, but it's not just there as a soluble.
Yeah, the activity goes down by 100-fold if you don't have it incorporated into a membrane, and it's basically localizing the clotting reactant cell surface. And there is a different form of tissue factor called alternatively spliced tissue factor that's soluble, and that doesn't have meaningful activity. But if you do antigen-based assays, then potentially if you do that in plasma, you can pick that signal up, which again is not really what we're interested in. I think the other comment is that if tissue factor is on a vesicle in plasma, it will bind 7A, and we've got evidence that it's also binding to some degree the inhibitor, tissue factor pathway inhibitor, which sort of makes sense because you're going to block that. So if we put an antibody against TFPI in the assays, we do increase the level of EV tissue factor activity. One of the bead capture assays also found that there was TIFB binding to the bead, and they concluded it was TIFB binding to the bead, but we think it's TIFB binding to the tissue factor 7A complex on the vesicle, and we're currently doing some studies on that.
Yeah, so that's going to the question then about TIFP and thrombin generation. So thank you for that. And Jayachandra, and if you're watching this on YouTube, please feel free to reach out to the authors too if you have any further questions about that. And I think what I just heard from you, Nigel, so there's so much good information that's coming out. So the tissue factor, if it is somehow cleaved or dissociated, you don't have that activity, or at least the same level. So that's why it's so important to be looking at this, the activity levels, not just worrying about where it is exactly, because that's actually correlated. We have, so I see that. Let's see, Lucio, go ahead. Yeah, so you have your hand up. Then we'll come to Alain.
Hello, I have a question. I think it's a question for Anna, but also for Nigel. So your studies goes in pretty much in the same direction of other studies by measuring activity instead of antigens. So must be some correlation between activity and level of expression of this tissue factor, don't you think? And is there any antibody that you believe in, or it's everything about of different isoform, different cryptic or uncryptic isoform of the tissue factor?
Could I just clarify the question for Anna? You're saying, is there a correlation between expression and activity or antigen and activity? Wasn't entirely clear which of those two.
So the procoagulant activity is increased in some patient with some inflammatory disease. Do you think there is a correlation with level of expression, or we cannot believe in it in any way? Is there a way to correlate the level of expression of tissue factor and its activity in somehow?
Okay, so if I understand the level of expression, then would be in the, for example, in the EVs, is that right? Okay.
Yes.
So we actually tried to look at the tissue factor levels of expression or antigen in these EVs, and we tried several commercial ELISAs, and we didn't have good results about that, but only when tissue factor levels were really high in these plasma samples, then we had at least a trend of association between the levels of antigen and activity in the EVs. So it could happen, but still we didn't have a very good method to analyze the tissue factor antigen in EVs. So that's something also that we're.
So what is increased in this patient with, for example, COVID? It's the number of EVs expressing tissue factor, is the amount of molecules tissue factor per unit of surface on the EVs.
Yes, it's the number of tissue factor positive vesicles are increased in patients with COVID, cancer, etc. I think the other important thing to point out for people who don't study tissue factor is tissue factor comes in two flavors. One is an inactive tissue factor that has no activity. It still binds 7, but it's got no activity. And then there's a second flavor that's active. And there's a series of different mechanisms that can take an encrypted inactive tissue factor to form an active tissue factor, and one of which is this phosphatidylserine flipping from the inside of the membrane to the outside of the membrane, where it can interact with the tissue factor, change its conformation, and then turn it into an active state. So again, one of the issues with measuring antigen is you actually don't know whether that's an active or inactive tissue factor, whereas if you look at the activity, then you know it's the active form of tissue factor that you're studying. And there's other forms of regulation of the tissue factor that determine whether it's in the encrypted or decrypted state. But that's why we prefer to look at the activity rather than antigen, not only based on sensitivity, but also on functionality.
Excellent. All right. So we have a comment here from Alain Bresson. Alain, would you like to tell us a bit more about ultracentrifugation? So sometimes we hear we should never use ultracentrifugation because it's going to do this and this and this to you.
Well, hi everyone. Great presentation. Yes, I want to make a comment after what was said earlier by Young about says some people think that ultracentrifugation may lead to some structural changes, damages to EVs. Well, this is not correct. I think cryo-EM is the best way to look to get the structure of EVs. And contrarily to Nigel, I do care about EV structure. And we've been looking at many different types of EV samples. And I mean, EVs are not so fragile objects. In fact, they are very resistant. And what occurs upon high-speed centrifugation is some aggregation of EVs. But by the way, not all EVs do aggregate. Some prefer joining other EVs and others don't. But the extent of aggregation is very difficult to quantify. You may see some by flow cytometry, for example. But basically, I would say ultracentrifugation will not damage that much your EV samples. On the other hand, I would very not recommend using some kits of EV purification, because with some of those kits, you have huge damage on the EV structure. I'm not going to give you the names because I don't even I've forgotten the names. But well, anyway. Well, that's about it. And I may say that I've been trying to see an TF EV by immunoGO labeling for days. I spent days and weeks behind the microscope with many different antibodies. Not a single positive result.
Yeah, wow.
I'm not totally abandoned, but perhaps one day I'll go back to that. Well, thank you very much for the for the presentation and for these meetings. Okay. Bye.
Yeah, that's there's some very good comments, and we always have to remember that our separation methods, that every separation method that's out there has some sort of a purpose, and a good use, and we just have to keep in mind the positives and negatives of those. And the more that we understand about the separation procedure, which unfortunately in the case of kits, we often don't know anything about it except the basic principle, the more that we can add to interpretation of our results. Let's see, we're coming towards the end of the hour, but we have another question here. So we've had some good back and forth, but most of the time we've been talking about blood. And Brink, you have a question about some other biofluids.
Yeah, it's just a more general question to Anna and Nigel. I mean, we published some recent studies on milk and saliva and tears and urine even, and we find lots of vesicles with tissue factor. They're extremely coagulant. I mean, easily 100 or 1,000-fold more active than what you measure in blood. So do you dare to speculate why normal people, as we are all listening to this talk, why do we have these vesicles with such a huge tissue factor activity? And in fact, just as a comment also to Ellen, we also recently published that, for example, in normal human milk, if you isolate the vesicles, the tissue factor is already complex with factor 7. So perhaps that may also explain why it's difficult to see it on cryo-EM. Because perhaps factor 7 is blocking the binding of the anti-tissue factor antibodies. But do you dare to speculate why we have so much coagulant tissue factor activity in normal human milk, saliva, urine, tears, whatever, all associated with vesicles? Why?
Nigel?
You want to take a shot? So this comes back to the 87 paper from Tom Drake, which is a beautiful paper, if anyone's not seen it, in American Journal of Pathology. And he coined the term that tissue factor produces a hemostatic envelope, and that surrounds blood vessels, and that's obvious why we would want to protect and maintain hemostasis around blood vessels. But it also is at every body surface, particularly the epithelium and the skin. And so if you have an epithelial cell producing a lot of tissue factor, it doesn't surprise me that it will release some of that tissue factor into that fluid. And I think that would then function as providing additional hemostatic protection to the cellular tissue factor, so that when you had damage, you could then recruit that vesicle tissue factor to help stop the bleeding at that site. And that really makes a lot of sense to me. So it's an extension of Tom Drake's 87 paper, where he was talking about the cellular body surface tissue factor, and then we are going to add an extra layer to that, which is the vesicle tissue factor being released by those cells.
Thanks.
All right, we had two more questions that have just come in. I'm just going to summarize these quickly. So we have a question about the large and small EVs, and how did you normalize input into your activity assays?
Okay, so in the activity assays, we just centrifuge the samples, the EVs, and we're suspending the same amount of the initial volume of plasma, which is 100 microliters, and then divide this into 2 wells to do the incubations with or without the antibodies. So it's based on the volume and not concentration of EVs.
So the volume of the source. Very good. Okay. And then finally, I don't know if we're going to be able to answer this today, but Wolf is asking, is it possible to see the protein corona on EVs in cryo-EM?
Yeah, I'm sorry, but I think this is one of the upcoming topics, the protein corona on the EVs. And I think also tissue factor might be associated with this. So depending on the isolation method, if you use SEC or TFF or ultracentrifugation, and Alain just told us it doesn't matter that much, so it doesn't affect the integrity of the EVs, but does it affect the integrity of the corona?
Mhm.
Do you believe in Corona?
Yes, I do.
I don't.
Okay.
Shall we have a, we should have done a poll. Do you believe in the corona here? But anyway, it's too late for that today. But certainly this seems to be a very hot topic right now in the field. Different findings on how closely different factors are associated with EVs. And so I'm sure we'll have plenty more opportunities to discuss and debate on the corona. But thank you everyone for your participation today and for all of the excellent questions. It's been a robust discussion. I've certainly learned a lot. And special thanks to Anna for your great presentation, for all the work that you put in here, to Nigel for supervising this work and for really advancing tissue factor research. And Rink, thank you too for joining and for suggesting today's topic. So take care, everyone. Hope you have a great rest of the week, and I look forward to seeing you again soon. Bye now.
Thank
Thank
you.
you.
Thanks.